Glycerol is the backbone of the phospholipid. It has 3 carbons with a hydroxide on each. These hydroxides are used in dehydration reactions to attach other molecules. Fatty acids are carboxylic acids with a variable length straight chain of carbon-carbon bonds. These acyl groups can be from 14 to 26 carbons, but are usually 16, 18, or 20 carbons for mammals. Phospholipids have 2 fatty acid acyl groups attached to the glycerol backbone by esterification reactions. This forms diacylglycerol, which can be a second messenger. Phosphate is an important inorganic molecule in cells. It is attached to the 3rd hydroxide of the glycerol backbone by a phosphoester bond. This forms lysophosphatidic acid. LPA is a signaling molecule released by apoptotic bodies. Phospholipids are made by esterifying polar molecules to the phosphate. There are 4 main phospholipids that differ by the polar head group: phosphatidylserine, phosphatidylethynolamine, phosphatidylcholine, and phosphatidylinositol. Phosphatidylserine is a Zwitter ion, having both positive and negative charges at cellular pH. It is normally restricted to the inner leaf of the membrane but is flipped to the outer leaf when a cell undergoes apoptosis. This is an eat-me signal for macrophages. Ethanolamine is serine without the acid group. This phospholipid is phosphatidylethanolamine. It is an important phospholipid during autophagy. Choline has a quaternary amine group and therefore has a positive charge. Phosphatidylcholine is the most prevalent phospholipid, representing 45 to 50% of the membrane. Inositol is a cyclohexane ring with a hydroxide on each carbon. When esterified to the phosphate, it forms phosphatidylinositol. The numbering of the carbons in the inositol rings is important and begins at the esterified carbon. Carbons 3, 4, and 5 can be phosphorylated. Each phosphorylation is important in a different context. Phosphorylation at the 4 position produces phosphatidylinositol phosphate. Phosphorylation at the 4 and 5 positions produces phosphatidylinositol 4,5-bisphosphate. This is very important to signal transduction, as cleavage by phospholipase C at this position produces the 2 second messengers IP3 and diacylglycerol. Phosphorylation at the 3 position, in addition to the 4 and 5 positions, produces phosphatidylinositol trisphosphate, or PIP3. This is a binding site for proteins with a plextrin homology domain, and it brings these proteins to the plasma membrane during signal transduction. Finally, phosphorylation at only the 3 position produces phosphatidylinositol 3-phosphate. This is an important signal during autophagy.